There we go. Briefly, I'll talk today in my part about the program. And just to remind you, what is the goal of this program and what is this program actually going to be focused on? So we're going to talk about some of the tools, methods, and data repositories. We'll talk about some of the curated examples and peer-reviewed publications that you should try to go through during this program. We'll go through some tools so that you can start analyzing data and you know where to find the relevant resources. And importantly, we'll talk about the relationship between how data is generated, how it is processed, how it's analyzed, how it's visualized and presented to describe a specific infectious disease, or potentially how it develops and what are the outcomes. We'll also talk about how, as a part of this program, hopefully you will have the opportunity to develop an independent research project and then present it to some mentors for feedback. So the program will talk about tools that are commonly used, and we'll focus on that first. So we'll talk about things like sequence variation, how to align sequences, how to characterize the variation. We'll talk about evolutionary analysis and how to link mutation rates and time. But we'll also talk about the specific applications of those in different domains, and importantly, subsequence variation, so subconsensus variation, sorry. We'll talk about functionality and protein structure, how a lot of the sequences that we'll be dealing with are protein coding sequences, and that's why we can explore interaction between different proteins and try to understand what can we learn about the impact of a particular variation on the function, on how those interact. And then we'll also talk about transcriptomic data analysis, because when we think about the infection, the viral or bacterial genome is what kind of characterizes that infectious pathogen, but we're also interested in the human response or host response, and we can characterize that really well with transcriptomic data, including bulk and single-cell RNA-seq. So we'll talk a little bit about that as well. Today, though, what are we going to try to achieve today? So first of all, I will kind of use this pandemic to talk about bioinformatics in infectious diseases, and we'll talk a little bit about how this pandemic really highlights the application of bioinformatics tools, but we'll also talk about how it guides the development in population health, how people are thinking about epidemiology, and also the development of vaccines and antiviral drugs. And we'll talk about how this online program can work in the best way possible for you, based on your background, based on your interests, based on your objectives and goals. Where can you find the necessary resources for you to be successful, and what can you expect to get out of this program? So, as Sonarik already mentioned, the central location where you will find all of the resources that you need is going to be on this OmicsLogic portal. And what we'll do first is we'll make sure that everybody is set up to do everything that you need to do throughout the program by completing your sign up and your profile. So how many of you already have a completed profile? All right, Anu, did you complete your profile already? Okay, let me show you what a completed profile looks like. Then we will jump into our exercises. So here, if I log in to learn-omicslogic.com, you can see that I'm logged in. I can click on my name, and that will be my profile. So you should be able to recognize me. Hopefully I look as beautiful as I do on this profile picture. If you want to connect with me, you're welcome to connect with me on Twitter, on LinkedIn. Also see my org ID right here. And what I can do is I can share with you my profile just by clicking right here on top and sharing my URL. So as we're going to be going through this program as a team, as a group, let's make sure that we have all of these profiles completed. If something is not complete, you can just click on your profile, and right here you can add all of that information. Make sure that you include the full link to your account if you want to share your social. And if you are not comfortable with uploading your own picture, you don't think it looks that great, you can just put a logo or maybe some picture, and that will help us see that you've completed it. Actually, it's going to be important for you to be able to work on an independent project. All right, so I see that Clement has completed, and I understand Anu also. Did anyone else? All right. Anaga, thank you. If you have, please share the link to your profile. So we'll save that for later. We'll be able to follow up. All right, briefly, I'll show you what you can do with this profile. You'll be able to go through different courses as we go through the program. And these courses are going to be related to different topics that we cover. For example, infectious pathogens like malaria, EVT68, SARS-CoV-2, all of them have an associated publication with a data set with some tutorial that you can go through. And that is a part of this curriculum. So as you go through and you want to go back to where you left off, sorry, you can always see that information right here. Your objective throughout this program is to get to 10,000 points. So you can see that I've been doing this for a while. I have over 26,000 points, but all you need to do is actually get to 10,000 points, and 10,000 points will allow you to work on an independent project. So that project here, I have some demo examples, but you'll be able to actually start a new project, and this is like preparing for a publication. So you'll be able to use this as a poster presentation. We, by the way, have an online virtual symposium at the end of the summer that you're welcome to participate in that. But really, how do you demonstrate that you've learned something in bioinformatics for infectious diseases? The answer is you do it by showcasing your work. And showcasing your work also provides you with the benefit of connecting with mentors and experts in the field that can comment and suggest improvements to your project. So this is a critical component of this program, and I'm really hoping that you will be able to do this. Now, if you have a question throughout this program about the data sets, about the right kinds of research questions, all of that information, we actually have a course on that. And so one of the requirements for anyone who would like to complete a research project is, number one, to have a completed profile. Number two, to get to at least 10,000 points. And number three is to complete this course, course 9, Designing a Bioinformatics Research Project. So why is this so important? This is important because our objective throughout this program is to help you learn about bioinformatics and in application to infectious diseases, but also to acquire practical skills. And one way that we can know that you have acquired those practical skills is, of course, to ask you. But if we just ask you and you tell us, there's really no depth to that understanding. So that's why we design our process in the learning of omics data and analytics by structuring our content from basic, understanding the terminology, understanding examples, understanding what we're talking about, all the way to this independent thinking and ability to construct an analytical process by understanding what analysis matches what kind of data, matching what kind of question. And all of those things are kind of integrated on this online portal. So here's just some example of recent graduates who have completed our program. You can see that a couple of them have been able to cross that 10,000 points, but not everyone has. And that's okay, because really the objective here is to fit the program to your needs and not the other way around. I hope that all of you would be able to complete an independent project, but if you're not ready for that and you need more time, that's okay as well. But I do think that at least you should be able to understand the terminology at the end of this program and showcase what you have learned. And that's why we have certificates for each course that you will complete. We have examples, so you can see how these methods are applied in different types of infectious disease studies or experiments. And you will be able, you'll be able to at least do some literature review and kind of read through the methods of these publications and really understand what others are doing. So before we go on, I'll introduce myself and another mentor that you will work with throughout this program, Dr. Raghav Lakshminarayan. My name is Ilya Brodsky. Really, I'm a manager, but I do have bioinformatics experience. My background is in applied mathematics and data science, and I'm currently doing my MBA. But really, I want to tell you about the reason why I'm interested in bioinformatics for infectious diseases. And then if anyone would like to share a little bit more about your background and why you've joined this program and what your goals are, I'll kind of, allocate some time for that. So right before the pandemic, I was invited to participate in a project that was financed by DARPA. And actually, before the project that I participated in, there was another project that we kind of applied for a grant funding to participate in, and that project was called Prophecy. Now, Prophecy sounds like, this interesting name. Practically speaking, what they were trying to do is they were trying to develop a combination of experimental techniques and analytical and mathematical modeling techniques to be able to predict which viruses might break out, might cause an epidemic or a pandemic, and how that pandemic is likely to kind of spread. So that original project I thought was, very fascinating idea. We applied for funding. We kind of, thought about different types of tools and methods and mathematical modeling that can help us in that. Ultimately, we did not participate in that original project, but we reapplied again in the next term. And that project was focused on a therapeutic prevention of such an outbreak. So remember, this is all before the pandemic started. No one is thinking coronaviruses. No one is thinking COVID. But everybody in the group, these were people from Stanford, from UCSF, from NYU, from all of these great medical research centers. And everyone was proposing their set of viruses. So there were people that dealt with Ebola virus and with Nipah and with all kinds of HIV and other types of epidemic and pandemic type viruses. And as I saw them interact, it really drew me into this whole topic because I realized that we kind of live in a world where viruses, bacteria, pathogens, they are out of the sight until something really terrible happens. And we don't, all of these people, they were complaining about lack of funding and lack of, focus and attention to what they were developing and lack of kind of public health policy about these infectious diseases. And right towards the end of that project is when this pandemic actually started. And that was just amazing to come into this pandemic with so much understanding of actually what's going on, what kinds of tools and methods and ongoing research is going on. And it really allowed me to appreciate the work that a lot of these research scientists were doing at the time. And I even remember after the pandemic kind of started to subside, I went back to the gym. And one of the people that we worked with on this project became kind of a more prominent public figure speaking about the pandemic and the research that they were doing at University of Pittsburgh. And I kind of knew the guy, we worked together and things like that. So it was really an interesting paradigm shift for me going into that project and then eventually kind of seeing how the pandemic unfolded and is continuing to unfold. So that's my personal story of how I got interested in bioinformatics for infectious diseases specifically. Is there anyone else that would like to share kind of, why did you think to join this program now? And what would you like to achieve as a part of your training? I'll just give it a moment. If anybody wants to, I'd love to hear. If not, that is also fine. Yes, go ahead, Halkano.
Yeah, my name is Alkano. I come from Kenya.
Mm-hmm.
I've been following a couple of YouTube webinars from Pine Biotech, and I've seen the quality of the work that they are representing, which is in line with what I really want to pursue in future in terms of applying bioinformatics in infectious diseases. My background is in chemistry, and I really want to, at some point, get experience also in looking at the aspect of drug discovery in, for instance, malaria.
Mm-hmm.
Yeah, because in tropical countries like sub-Saharan Africa, infectious disease is a public health concern.
Mm-hmm.
Yeah, so I want to work at the interface of looking at novel drugs that are sourced from natural products, and now understand that from bioinformatics point of view.
Excellent.
And I've seen that I might get that kind of skills skill set from Pine, from this company.
Excellent. Yeah, thank you, Halkano, and definitely a very concerning and a big topic to address there. So thank you very much. Hope that you achieve your goals, and we'll probably have a chance to interact more about that. So let's give a chance to Clement. Clement, would you like to share your background and how you'd like to use this time?
Yeah, greetings from Nigeria. My name is Clement. I fell in love with bioinformatics after attending a series of webinars, like my colleague from Kenya just said. I came across this training in one Telegram group, and I appreciate the administrator for the WhatsApp group too, who decided to mentor me on this. After about 29 years of bench work, I just took up academic appointment, and one of the topics listed by the faculty is bioinformatics. So I really look forward to learning the intricacies, the applications, the foundation of bioinformatics. Thank you very much. It's nice being with you all. Thanks.
Excellent. Excellent. Thank you, Clement. Thank you very much. Okay, great. So again, we'll have more opportunity to connect with each other and get to know each other. So please, continue to share your links to your profile. That's really, I think, the easiest way for us to connect in the beginning. But let us switch gears, and we'll briefly talk about some of the tools that we're going to be using in this program. And I want to give you a little bit of an illustration today as well, so you kind of get a sense of where we're going, what might be the potential outcome towards the end, what you would acquire as a result of going through this program. So we're going to be using these three primary resources. One of them is going to be the OmicsLogic portal. And as I showed already, and Sonanika showed in the beginning, we will have project examples, curated data sets, some training materials that everyone will have the opportunity to go through. The second portal is going to be the Bioinfo platform, and has some dedicated sections designed to facilitate the types of studies that we will go through. And those include processing, analysis, visualization of different types of analytical methods like multiple sequence alignment, phylogeny, transcriptomic data analysis, integration, machine learning, and others. And then we will also learn a little bit about coding in both R and Python. Now, coding is like a language, and once you've started, you can really start speaking about anything that you would like. But one of the important parts of coding exercises that we will do in this program are going to be related to the methods that we first cover on the platform. So you understand the logic first. Why are we doing what we're doing? How do we get from A to B? And then we will look at typically some packages that somebody already prepared, and we'll focus on the application. So we're not going to spend too much time on the algorithmic part, the mathematics of these methods. But if you would like to, you're welcome. We will share some publications and some links where you can read more about all of those methods. Okay, so again, we don't have that much time left, but what first I want to do in this first part of this presentation is briefly talk about some of the traditional and novel methods in bioinformatics used for infectious disease research. And I want to give you some examples that include working with high throughput sequencing data, how to detect and characterize a viral strain, and also the relationship between nucleotide and amino acid, and the mapping of sequence variation on protein structure. So these are some things that we'll kind of go through